Our next adventure awaits us at the interactive kids zone inside Epnot Center. Let's take a quick glimpse inside while we cover the DNA repair mechanisms. Before we start, you may want to check out our DNA mutation sketch if you haven't already. All right, so what's DNA repair? Well, it's a series of events where enzymes recognize and fix damaged or mispaired DNA. If that damage isn't identified and fixed immediately, mutations accumulate inside cells, leading to all sorts of clinical problems down the road, like cancer, which, by the way, is bad, in case you were wondering. Got your attention? Good. Let's go over the DNA repair pathways. There are 3 ways to repair problems with a single strand of DNA: nucleotide excision repair, base excision repair, and mismatch repair. Nucleotide excision repair, or NER, removes bulky helix lesions, like pyrimidine dimers, that form due to UV radiation and distort the DNA double helix. This repair pathway takes place during the G1 phase of the cell cycle. In the first step of the repair, helicase unwinds the damaged double-stranded DNA after the pyrimidine dimer is recognized. All aboard! We'll use our recurring helical coil train to represent helicase. See the little tool it uses to unzip the tracks? Next, endonuclease creates nicks in the DNA backbone to remove the damaged nucleotides. This kid's safety scissors will remind you of endonuclease. Braden? I don't think you're supposed to shred the exhibits, bud. Then DNA polymerase fills the gaps with correct, undamaged nucleotides. And finally, ligase seals the gap by linking the DNA backbones together. Just like that kid fixing up after Braden's reign of terror. This is a good time to mention xeroderma pigmentosum, or XP, an autosomal recessive genetic disorder caused by mutations in the NER enzymes. Affected patients are unable to repair DNA pyrimidine dimers after being exposed to UV light. As you might expect, XP primarily affects the skin. The key findings are hypersensitivity to sunlight, severe sunburns, corneal ulcers, and skin cancer. Anyone with xeroderma pigmentosum should avoid sunlight exposure. If they must go outside, they should wear protective clothing and use liberal amounts of sunblock. All right, let's transition to base excision repair, which we'll represent with this definitely stylish, totally in fashion beret. As the name suggests, BER removes single bases that have gotten messed up. In particular, it can fix deaminated base lesions, as well as oxidized and alkylated bases. Toxins and carcinogens cause these base lesions to develop in the first place, and BER can be activated any time during the cell cycle. In the first step of BER, glycosylase identifies and removes the base lesion to create a so-called AP site, which contains just the phosphodiester backbone. AP stands for apurinic or apyrimidinic, which just means there's no nitrogenous base there anymore. Let that kid enjoying a sugary treat and taking out gross bits of track remind you of glycosylase. Next, AP endonuclease cuts the 5' end of the backbone to make a little nick, while AP lyase does the same on the 3' end. Uh-oh, more troublemakers cutting up track. We'll represent endonuclease with scissors again, while lyase is that girl cutting the track while lying down. The final 2 steps are pretty much the same as NER. DNA polymerase fills the gap by adding new nucleotides at the AP site, and ligase seals the DNA backbones together. The final single-strand repair mechanism is mismatch repair, or MMR, which sounds exactly like what it's supposed to do. MMR identifies and replaces mispaired bases that form during DNA replication, and serves as the primary backup repair mechanism when DNA polymerase misses errors. As such, MMR takes place during the S phase. We won't draw each step here because they're pretty similar to NER and BER. First, mismatch repair enzymes identify the mispaired base in the newly made unmethylated daughter strand. Next, an endonuclease creates a nick in the daughter strand and removes the mispaired base. Then, DNA polymerase fills the gap, and finally, ligase seals it. Okay, time for a clinical pearl. MMR is defective in Lynch syndrome, also called hereditary non-polyposis colorectal cancer, or HNPCC. Illustrated here is a inchworm toy to remind you of Lynch syndrome, an autosomal dominant disorder. At Sketchy, autosomal dominance is represented by our recurring domino pattern. Patients with Lynch syndrome usually inherit a mutation in one of the genes responsible for MMR, while the second allele undergoes spontaneous mutation. As a result, affected patients will have a higher likelihood for developing colorectal cancer at an early age. So far, we've covered the single strand repair mechanisms. But what about double stranded DNA? Well, there are two pathways to correct double stranded DNA breaks: nonhomologous end joining and homologous recombination. Nonhomologous end joining, or NHEJ, glues the broken ends of double stranded DNA without using the homologous chromosome as a template. Remember that homologous chromosomes are chromosome pairs inherited from each parent. To remember NHEJ, picture this kid joining the ends of the tracks. Since NHEJ doesn't use a template, whatever is put back together might miss a piece of DNA. So, yeah, expect more errors. Time for another clinical pearl. Ataxia-telangiectasia, or AT, an autosomal recessive disorder, is caused by NHEJ defects in the ATM gene. AT presents with cerebellar ataxia, telangiectasias, and increased risk for respiratory infections. The other way to repair double-stranded DNA is homologous recombination. HR uses the complementary strand of the undamaged homologous chromosome as a template to fix the double-stranded DNA. With that in mind, HR has a much higher accuracy than NHEJ. Besides repairing double-stranded DNA, HR promotes genetic variation in offspring by mixing genetic material between homologous chromosomes during meiosis. Okay, one last clinical pearl for you. Hereditary breast ovarian cancer syndrome associated with BRCA1 mutation and Fanconi anemia are caused by HR defects. Look, kid, if you're mad about having to eat broccoli, yelling into the breeze ain't gonna help. I suggest a sternly worded letter to your parents. Whew, well, I think I'm finally finished cleaning up after Brayden. How many times am I gonna have to ban that kid? Let's just do one last check to make sure I got everything. DNA repair is a process where enzymes identify and fix damaged or mispaired DNA. NER removes pyrimidine dimers made by UV radiation. After the pyrimidine dimers are identified, helicase unwinds the DNA. Next, endonuclease removes the damaged DNA before DNA polymerase fills the gaps and ligase seals the nick. BER removes deaminated, oxidized, and alkylated base lesions. First, glycosylase removes the base to create an AP site. Next, AP endonuclease and AP lyase create nicks in the strand before DNA polymerase fills the gap and ligase seals the nicks. MMR removes and replaces mispaired DNA with the correct bases. NHEJ attaches the broken ends of double-stranded DNA without using a template, leading to more errors. HR accurately repairs double-stranded DNA using a template and promotes genetic variation in offspring.